cloud. All right, take it away.
All right. Hi, everyone. Thanks for letting me talk to you today. This is just a kind of an overview of a lot of the stuff that I did during my PhD and some of the stuff that I'm attempting to wrap up now for publication. So I work in Cnidarians. I think most of us here are marine scientists or biologists and understand that Cnidarians are a phylum of animals that include our jellyfish, our anemones, and our corals, and also other animals such as hydra and hydractinia. They are classified by the presence of cnidocytes, or stinging cells, in their tentacles. So these are the things that we associate with a jellyfish sting for this particular phylum. So this is just a really nice blanket overview of photos of different animals that fall underneath this classification. So I work on something called the Nigerian algal symbiosis, which is a symbiotic relationship between dinoflagellate algae and some Cnidarian host. So the host on this page is Cassiopeia, the upside down jellyfish. We kind of go into a zoomed in view of the tentacles, and you can see the little round algal cells up top. And then below that is a picture of the Cassiopeia oral arms, and all the little brown dots you see there are algae. This symbiotic relationship is classified by the exchange of nutrients. So the algae photosynthesize, those photosynthetic byproducts are passed along to the host, and they make up, it can vary depending on the host Cnidarian, but it can range from anywhere to up to 70% of the host nutrition coming from these photosynthetic byproducts. And in exchange, the algae get protection from predation and other nutrients that they need to perform photosynthesis. So we can talk a little bit more about the algae in detail. My current research really focuses more on the algae versus the host. So this is a family called the family Symbiotidaceae. It used to be a genus called Symbiodinium. And about 3 years ago now, it was split into various genera. Each genera was split into multiple species and or strains. So again, on the bottom here, we have some close-up images of the algae themselves. And on the side, this is the tree that came from the 2018 paper that shows the old clade names along with their new genus names. And so again, under each genus, you can have an abundance of strains, and each strain can have different characteristics. So the thing that I'm most interested in when I'm looking at Cnidarian symbiosis is understanding bleaching, and bleaching is usually correlated with corals. So coral bleaching is the expulsion of the algae from the host due to some sort of stress mechanism. So this is one of my favorite schematics. It's from NOAA, and it shows the stages of bleaching. So in the panel one, you have a healthy coral, and as you saw with the Cassiopeia slide, those algae give a kind of brown coloration to the animal. So in a healthy coral or a healthy symbiotic Cnidarian, you'll kind of have this brown coloration. Under some sort of stress, typically we're talking about heat stress due to climate change. So this symbiotic relationship is really happy under a certain temperature range. If you go above that, the symbionts or the algae start to lose their photosynthetic function, so they start to break down. They're no longer of use to the coral or the Cnidarian host, and the host begins to expel them in large quantities. So this is when we get bleaching. So we call it bleaching because once the algae are gone, the host no longer has that brown coloration. They turn white, bleaching. So I like to give also kind of a blanket overview of how bleached animals look across the Cnidarian phylum. So we have on the left kind of your standard coral bleaching here, some acropid corals at the top. At the bottom, a bleaching event that happened across the American Samoa in under a year. In the middle here, this 2006 photo is a sea anemone called Anthopleura that was bleached in a laboratory setting. In the top right-hand corner, we have Aptasia, which is one of the most popular model systems for studying symbiosis. That's been bleached. Below that, a broom coral, And then under that, again, we have the upside down jellyfish, Cassiopeia. This is my photo from a experiment that I did during my PhD, in which the Cassiopeia was bleached under heat stress. Okay, so we can talk a little bit about Cassiopeia, which is my main model system. I started working on Cassiopeia as a PhD student. I still work on Cassiopeia now, in addition to some other symbiotic cnidarians, but this is kind of what started my career and what most of my experiments are formulated around. So again, Cassiopeia is a cnidarian. It's a scyphozoan jelly, so what we think of as a true jellyfish. It is also called the upside down jellyfish. They live upside down in mangrove beds because they have their tentacles pointing up towards the light, so those algae can photosynthesize. And they're kind of an up and comer in the symbiosis world when talking about model systems. So as I said, Cassiopeia is a jellyfish. It has a typical scyphozoan life cycle. So we have two sexes, separate sexes in the medusa stage. These produce what we call a aposymbiotic planula. So aposymbiotic planula just means that these planula are not infected or inoculated with algal symbionts. These planula will kind of float around in the water column for a little while, then eventually settle on some sort of substrate, could be a mangrove leaf, a rock, things like that. They will grow up into the aposymbiotic polyp stage. So again, at this stage, there aren't any symbionts associated with the animal. During this stage, the animal can actually asexually bud to produce small clones. These asexual buds behave very similar to the planular larvae, in which they settle and grow and produce another polyp. At this stage, at the polyp stage, is when the animal will acquire symbionts from the water column. So they take them in during feeding. So inoculated with symbionts. Cassiopeia, this is where Cassiopeia starts to differ from other scyphozoans, in which the only way that Cassiopeia can move on through the rest of the life cycle is if they're inoculated with symbionts. So once they have a high enough complement of symbionts, the animals will strobulate, go through a process called strobulation, which is really common among scyphozoan jellies. So the top portion of the polyp where the tentacles are will pinch off to form what is known as an endophyra or a juvenile medusa. The endophyra will continue to grow into the medusa stage that we started the life cycle with. Okay, so that was just kind of an overview. I wanted to talk a little bit about the model that I'm using for these experiments. I typically focus on the, go back a minute. I typically focus on the polyp stage when I'm doing experiments. So that was just an overview of understanding the system that I'm using to infect the algae that I'm going to talk about next. Okay, so the work that I'm doing now is working in more of a cell biology lab. I'm trying to understand the Nigerian symbiosis. So in 2018, my postdoc advisor, Dr. Ting Zhang, she did work looking at trophic shifts of these algal cells. So typically in the lab, we will extract algae and grow them in a growth media on a light cycle. And this light cycle allows them to photosynthesize normally and grow and divide inside the media. She was working on a algal strain called SSB01, which was isolated from an Aiptasia species, which is what we have here on the left. These are some Aiptasia that she's working on. This SSB01, she found that she can grow it heterotrophically. So instead of growing it as a photoautotroph, she could supplement the growth media in the lab with glucose, which would allow the cells to grow without light. So they can grow in the dark and grow almost at the same rate, or actually at a higher rate than if they were growing as photoautotrophs. So this is a schematic here of that experiment. So it shows normal algae under a light cycle, and then they're a brown color. And then when they are supplemented with glucose, they actually maintain a bleached kind of look, or a lighter. They turn kind of pale brown yellowish. And that's because when they're growing in glucose, they actually lose their photosynthetic properties or photosynthetic functions. those mechanisms start to break down, so they are considered bleached at this point, but they're still able to grow and divide because they're basically feeding off the glucose that's in the media. This glucose bleaching is reversible. So if you take cells that have been supplemented with glucose, put them back into a normal media, they will regain their photosynthetic function and start growing as photoautotrophs again. Okay, so what we're doing currently is we're leveraging the ability of these SSBO1 cells to grow as heterotrophs to create mutant cells. So we work with collaborators at University of California, Riverside, Dr. Robert Jinkerson and his graduate student, Joseph Russo. They were working a couple years ago on creating mutants. So they were using UV mutagenesis, so UV light, to shine on these algal cells to create mutants. The only thing that they were finding is that these mutants, a lot of them had lost their ability to photosynthesize, so they couldn't survive in regular growth media. So then in comes Dr. Zhang was like, well, I've found a way to create, to allow these cells to grow without photosynthesizing in glucose. So now we're able to create mutants, have them grow in glucose media, and survive and grow to large quantities. So this is another schematic. So what we do is we'll place the algae under a UV light for a certain time period, depending on the level of mutation that you want to achieve before you reach cell death. So typically it's from anywhere from 30 seconds to 1 minute. So this is the SSBO1 Brevolium minutum, that's its species name. We will plate out the algae onto agar plates and allow what is there to grow up for a time. And then these mutants typically have some sort of different coloration. So again, the wild type cells are typically brown. These algal mutants can be green, yellow, orange, or really pale brown, white. So we'll use those color, that color specification to pick out the mutant colonies, place them into a glucose liquid media, and they'll grow up from there. And that's how we create our mutant colonies. So this is looking at some of the mutants that we work on that a couple of them I'm going to talk about for my work with Cassiopeia. And so we have this left panel is talking about their growth under as heterotrophs with the glucose media added, and then as photoautotrophs. So in that panel, you can see our wild type SSBO1, a mutant that we have that we call LBO1, and a mutant that we call GR7. And you can see, as I was saying, that they actually grow, and this is just streaked out on plates that are the plates for heterotrophs are supplemented with glucose, and the photoautotrophic ones are just normal growth media plates. So you can see at the under heterotrophic growth with the glucose supplement, they grow, the colonies grow a lot larger and a lot clearer. You can see the coloration a lot clearer for the mutant cells. So that supplementation actually helps their growth. And then on the right-hand side, we have another mutant. It's called Orange 1. And again, I didn't label it here, but it's the same idea. In this panel on the left, you have photoautotrophic growth, and then on the right, you have heterotrophic growth. And so again, a lot of these mutants have color variations, along with traits such as reduced photosynthetic function. Okay, so one of the questions that we wanted to answer now that we have a lot of these mutants that are deficient in photosynthesis is understanding, is photosynthesis required for the establishment of symbiosis in Cnidarians? So like I said, in Cassiopeia, these partners actually have to meet in the water column. So Cassiopeia is not, they don't, the planula do not come out with symbionts. They actually have to acquire them from the water column. And so we wanted to know if kind of the signals that go into establishing this partnership, if photosynthesis, if the photosynthetic function of the algae is one of those signals. So what we did is we took our SSBO1 wild type cells, our Orange 1 mutant and our LBO1 mutant, and we introduced them to a subset of aposymbiotic Cassiopeia polyps. So what we found here, so you can see in the middle panel, you can see all these red dots. So these algae actually autofluoresce red under certain fluorescent channels on the microscope. And so you can really easily see when an animal is infected. So what we saw here is that all these algae types, including the mutants, were able to actually form an initial symbiosis. So they were able to actually get taken up inside the polyp when introduced in the laboratory setting. The next thing we wanted to look at was if the animal actually went through strobulation, which I'll talk a little bit about next. So if they continue to go through the life cycle. So on this right side panel, I just have images of what wild type of fire red normally look like when the animal goes through a normal strobulation and a normal life cycle. So the answer here is photosynthesis required for establishment of symbiosis. And right now it seems that no, it is not. It seems that these algae can actually be taken up in large quantities without photosynthetic function being needed. So the next thing that I just said is photosynthesis required for symbiont growth within the host. So once they're taken in, will the cells continue to grow and divide and reach the quantity that's necessary for the animal to continue through the life cycle? So here we did another experiment where we took our mutant cells. So we didn't do the wild type. I did some separate experiments with the wild type only that shows that the wild type allows their animals to strobulate, which is important because these strains are all stemmed from that wild type strain. So I didn't show that here. But what we did is we inoculated another subset of polyps with LBO1, another subset with orange one. And again, we saw uptake. But over time, we were seeing that the cells were being expelled pretty rapidly from the host. So they're being kicked out. And these cells are not growing and dividing inside of the polyps. And we weren't seeing strobulation. So our conclusion here was that a small subset of cells can be maintained for a time, but there is no growth and there is no strobulation of polyps. So our conclusions is that is photosynthesis required for establishment of symbiosis, initial uptake? So the answer is no, it's not required. You can take up as many cells as you want at the beginning without them being photosynthetically functional. Is photosynthesis required for symbiont growth within the host? And the answer here is yes. Photosynthesis seems to be required, at least for Cassiopeia. If we talk about Aptasia, which is another system that I work on, that's a slightly different story. But for Cassiopeia, who is a Nigerian whose life cycle is dependent upon these cells functioning properly, photosynthesis is required for strobulation. And so that's pretty much it. This is something that we're trying to wrap up and publish, so be on the lookout for that. We used some other model systems to display the things that I was just talking about in Cassiopeia. So I would like to really thank the Zhang Laboratory, so Ting Zhang, who is my postdoc advisor, and all the students that work in the lab and helped me. Our collaborators, Dr. Robert Jinkerson and Joseph Russo at UC Riverside, and the Whitney Lab, which is where I did my PhD and where I was able to collect and maintain all my animals for both my dissertation and now. And you guys know how to contact me, so yeah. Are there any questions?
I have a question. Thank you for the great talk. For me, I am like the EE in the BWEEMS, the ecology and evolution. So I find marine science that very fascinating. I was introduced to coral bleaching maybe like 2 or 3 years ago. So I'm sorry, I'm a newbie to these things. But you mentioned like that. So first of all, it was a great presentation, laid it out well. Great figures, I followed it the whole way. I was curious, you said glucose made coral bleaching reversible. I didn't know that it could be reversible.
Well, I guess it makes the bleaching of the cells that we use reversible. Yeah, so I wish we could just go sprinkle some glucose. Like really easy and really great.
Okay, that was our next question. I was gonna say, why aren't they just putting glucose in the water? Okay, thank you. So is it a mutant or is it just a wild type in the lab strand that coral or glucose makes coral bleaching reversible?
We actually have some strains in the lab that I work on.
Oops, keep going. I'm good. It's my phone. It's my phone.
We can't bleach with glucose. So I work on a strain called SSAL1 as well. And that one we tried to grow on glucose, it didn't work. So we're looking now to see if there's another sugar that actually will maintain that as a heterotroph.
Okay, thank you.
Yeah, you're welcome.
You have a question in the chat?
Oh, okay. Let me.
I can read it out loud. So Nikki's asking, why do you think that they take up the defunct symbionts?
So that is a great question. So in Aptasia, it's actually really crazy because they'll like actually like hold on to the mutants for like a long period of time, which is a little bit different than what we see in Cassiopeia. I think in Cassiopeia, sometimes it can be like a passive uptake in a sense. But we tried it with a non-symbiotic strain, a symbiotic strain called SSE01, which doesn't form symbiosis, and Cassiopeia like didn't take that up at all. So like now we're like a little confused because SSE01 is part of this phylum. It is from Symbiotidaceae, but Cassiopeia was not interested in all. So there is some type of cue that they do take to pick out ones that are not symbiotic. But these ones, since they were formerly symbiotic, they take them in, but we're still kind of trying to understand that. So like, yeah, I'm not 100% sure.
So Nikki's asking, do we know anything about the immune cells in Cassiopeia?
No, so that is, I think, one of the areas that we are behind in terms of making this model system. No one formulates like immunity labs really around Cassiopeia. It's usually like Aptasia or another coral species. But that's something that I'm really interested in trying to like get off the ground one of these days. But yeah, so that's something we're kind of lacking in, unfortunately.
Let's collaborate, says Nikki. Okay, awesome. I'm actually very blown away because I'm an algal ecologist, and so I'm super like interested in this glucose being taken up by algae. We actually do phagotrophic assays in our green algal cells, so it's not the same as your symbionts, but very similar. And so I was wondering if there's been any studies on that particular algae where they eat heterotrophic bacteria or anything like that.
You mean like the microbial, like the microbes associated with these?
So instead of photosynthesizing, could they possibly take up organic nutrients from bacteria, or like, because we have green algae that actually eats bacteria in the absence of light.
Okay. Yeah, I don't know. I don't think that right now, at least what I've done, I haven't studied anything looking at bacteria with these guys. We actually really try to keep these cultures like super axenic.
Oh, good for you. I wish I could do that.
And so like that is because like the glucose media is really, really easy to get contaminated because of the sugar. And so like if you breathe in its direction the wrong way, it's like I'm contaminated. So it's like, oh, that's lovely. So no, we haven't done anything with bacteria just because it's so hard to keep everything really clean with those particular cultures.
Okay, awesome. Do we have any more questions? Looks like we're good to go. Thank you so much, Cassandra. That was such a good talk. I might reach out with more questions just because I'm so blown away by all of this. All right. So next up, we have Miranda Bernard from Arizona State University. She's doing her PhD there. Hold on, I have more information. She went to Princeton University and got her bachelor's in civil and environmental engineering. Oh my gosh. Did her senior thesis in irrigation and isotope analysis of crops in central Kenya. It's 7:30 in the morning and the coffee hasn't hit yet. I'm so sorry, everybody. And is currently at Arizona State University doing community-based work where she's interviewing folks in the Caribbean, specifically in a community in Trinidad and Tobago. So today she's gonna be talking to us a little bit about that. So Miranda, can you share your screen? Let me know if you have any issues.
You may need to make me co-host, or it says disabled.
Why? It worked for Cassandra.
Let me try.
Okay, try it now.
Oh, there we go.
Yay, we did it.
All right, yeah, so we're gonna be changing gears a little bit. That was super interesting, Cassandra, and I know very little about that type of work and those specifics of marine bio and ecology. Most of what I know is from teaching intro to marine biology, so that was interesting. I'm going to talk a little bit about like past work I've done, current work, and future work. So starting off, I'll kind of say how I ended up doing my research. as you said, my background is in civil environmental engineering. Okay, but I've always loved the environment, love spending time near marine creatures and at the ocean. My family is from Trinidad and Tobago, so I go there a lot and kind of got to see a lot of the intersections of communities and tourism and marine ecosystems. So this is going to be mostly pictures. So here's some leatherback sea turtles. The largest nesting site for them is on the north coast of Trinidad. And so you kind of like get a sense for how fragile and careful we have to be with our relationships to the environments. Like this is like a species in a nesting site where if someone decided to build some massive infrastructure to, you know, alter that beach coastline, that the like viability of this population would be completely altered. So I had moments like this when I was, you know, really starting to think more about like people and the environment. In my summers in undergrad, I got to do a few cool projects, just things by luck. Wasn't finding like internships in engineering, and kind of the opportunities that I was getting were all like marine ecology based. So this summer I studied blue holes in the Bahamas to kind of learn about like the past ecosystems there, and learning about how humans interacted with the environment, you know. So we found like pieces of charcoal, and so we could kind of date and see when were people starting to do burning activities on the island, when were there like large hurricanes. Yeah, so we got to do a lot of cool analysis there. And when I started grad school, you know, I love sea turtles, and I thought fisheries are really interesting. And I kind of thought, okay, well, let me try to study more about fisheries and bycatch reduction. And so I ended up at Arizona State University, not on a coast, but a lot of people do marine conservation here. And a lot of the research questions, I started taking classes in like the social sciences and learning about qualitative methods, and that started really sticking out to me. And I changed projects about 20 times in my first year and a half. And I kind of realized the more and more I thought about it, the more I was interested in these interactions with human communities and the environment. Specifically, I was interested in marine protected areas. And one thing that, you know, kept coming up in the literature is that, you know, originally there was a lot of research more on the ecology. Where should they be located? What size should they be? Should we do a lot of little protected areas as seascape, or should be one large one? But then over time, you see like this dialogue shifting towards, okay, well, it's not just an ecology problem. A lot of these protected areas we would think would be, you know, like successful in terms of their objectives, but they're not. So what's going on? And a lot of that has to do with the governance and the human dimension surrounding protected areas. And one thing that kept coming up is community engagement and the roles that can play in having like a successful MPA. But at the same time, there's not much information on like what about community engagement, because I think like we've all probably been engaged in some way as a community, and it's not always like a positive thing. So community engagement isn't inherently a good thing. It can lead to negative outcomes as well, because when we think of the word engagement, often we want to think of good things, but no, we have lots of bad engagement encounters, especially in governance or people coming into communities. And so I was kind of interested in finding out more about that. So reviewing the literature, you know, a lot of the engagement is very one-sided, and it's like one directional flow of information. And when we think about who's being engaged in the community, we also often think for protected areas a lot about fishers and business owners and people who have like this direct financial stake in the environment. But if you're in a coastal community, you have a connection to the environment in the space, and everyone's somehow connected to that protected area. So that was something that really stuck out to me. And this photo is Charlotteville, Trinidad and Tobago, where I conducted research on perceptions of environmental change, because that's also something, you know, people talk a lot about different sources of knowledge and perceptions being one of them, but they rarely are, there's not like that many case studies, and they're not fully understand on like why they're important and how they can be important, I guess is more so. So again, this is Trinidad and Tobago, so off the coast of Venezuela, I think like 5 to 10 miles. There's a lot of outflow from the Orinoco River, which is like a large river in South America. So there's a lot of nutrients in that water, and it's a pretty productive region. And so this bigger island is Trinidad, and the smaller one to the top right is Tobago. So zooming in a little bit, you can you see a lot of development in this southwest corner. And then there's one main road here, and as you travel along the coast, there's just communities all around waters or bays. And so Charlotteville is on this bay up here. And it's interesting to me because, well, the reason there's no development here is because this is all a protected forest. So it's the oldest tropical forest reserve in the world. I think it was established in 1776. So it's really old. And the reason it's established is because, you know, this area was colonized and changed hands many times by European countries over its history. And, you know, they had different crops, and from the like indigo, sugarcane, and something that they started to realize was they're transforming all this land into estates to have this agriculture, but they're realizing that without the forest, they're not capturing as much water. They don't have water to power the wheels to crush the sugarcane. So very early on, they kind of realized this, you know, connection to the environment, and then this forest was protected. I think it's just interesting to know like a little bit of the history and like why this forest is there. And so there's one road that cuts through it, so it's open to the public, and there's a lot of bird species, especially being so close to Venezuela over the past like thousands of years. There's been connection and disconnection to the South American mainland. So there's a very interesting mix of species on the two islands. So again, this is Charlotteville. And basically for this specific research study, I wanted to know about environmental perceptions over people's lifetimes and how those changed and differed. So I worked with a local organization there. This is another view. So you can see like this is all protected up here, and there's a fair amount of development, but it's kind of constrained because of that protected forest, and there's strict laws on who can build houses and how large they can be. So when talking to people about environmental change or changes over time, what we see a lot of is comments on the development and growth. So these are just counts of things people mentioned based into these categories on the left side. And so a lot of it's development growth, a lot about social dynamics. So how people are interacting with each other and how that social cohesion is changing over time, infrastructure like roads. And then we see like a few mentions of the environment and climate and weather patterns, and then a few about migration. But then when we think a little bit more about like development and growth, like we see that there's, you know, showing a lot of changes in the environment because As we know, when there's development, whether it's roads or buildings, that especially being right on the coast, that can lead to a lot of sedimentation in the water. And then, let's see, yeah. So then, like, here's kind of an example too of how things maybe used to be and is less common now. Is there used to be practices such as lent hand, where people would fish and they would help each other out and reel in these nets. But this is practiced way less now than it used to be. And it's more of like an individual system. So kind of there's changes in how we're interacting with the ecosystem based on these social dynamics that are changing, where there's less community, less ties between people. I mean, obviously still a lot, but just cited a lot less than it used to be. And then when we talk about environmental changes over time, it's interesting. We have a lot of people saying that there's no pollution, and a lot of people who mention that, part of what they were saying too is that there is pollution, but people pollute, but there's no pollution because there's someone whose job it is to clean it up. And so we may be like polluting the area, littering, things like that, but there's someone there to clean it up, government workers, so that there's really no pollution. So that kind of is interesting, and like a difference in how between some people in the community about how we're understanding pollution and what that means. One I'd like to highlight is yacht sewage. So there's not that much tourism in this town because it's pretty hard to get to, but what there is a fair amount of is tourists who come by yacht. And so they're really not interacting too much with the community. They kind of just anchor there for weeks or months. And, for the law, you're supposed to empty your sewage or dispose of things a certain number of miles offshore, but a lot of people in the community think that they're not doing that. And so that's really changing how people are interacting with the water, because a lot of people say, I'm not going in the water, it's like filthy, or it's not safe to go in. And so it's people who've used these bays for a long time, but now not so much, because there's this perception that there's a lot of yacht sewage in the water and that it's contaminating the water. But if you speak to people in the environmental NGOs there's kind of a disconnect, because they're saying that's not really happening, that's a problem. And then that's kind of where we see like the influence of the media, because it's something that has been mentioned in the media a lot. So whether we know it's true, it's kind of like, well, something has to be done because people think there's an issue and we need more information. And so that's not something that the environmental NGO is focusing on, but it is a big concern of people. So how can we address that concern when we're managing the environment? And then another thing that was like interesting was differences between men and women in the types of environmental changes. So women were more likely to cite changes that had to do with human health. So a lot of discussion about like air pollution and wastewater and in the yacht sewage, of course. Like on the right, you can see like these pipes going into the box drains, which then flow to the reef. And so this is kind of like gray water coming from the house. And so it's kind of interesting because, we're with globalization and everything, we have access to a lot more products, a lot more chemicals, when there used to be like one kind of soap that was more environmentally friendly, and that's what everyone used. But then there's all this trade and there's all this, influence and encouragement to use these other products, but how they're used in different locations vary. And then, they end up going into this box drain. yeah, so that's just kind of an overview of like one of my projects, but overall, I'm very interested in understanding how people in communities interact with the environment and how stressors and actions play a role in these dynamics, in these human environmental dynamics. And for future work, what I'm interested in is understanding threats to marine protected areas and coral reefs. And like we know, what's an example? Okay, so we know, I'll just use this one. So we know that there are invasive species that are problematic in certain areas. But then how does that actually impact people in the community? So what are these pathways that impact individuals in the community, especially individuals who we wouldn't normally associate with this like threat or stressor? And so understanding too, like then when we do MPA, like or whatever conservation interventions, how does that then affect people? Like if there's if you're going to change fishing quotas, well, what does that mean for people who work in the restaurants? Like, are there going to be fewer fish sold, and then are they going to receive fewer tips, or is it going to be a less valuable or economically valuable fish, and again, like less tips? And so how are these stressors to the environment actually impacting people in the community, and just kind of understanding those dynamics a bit more. And then this was just a photo I really like. Basically, they're dancing the cocoa, so they're on cocoa beans, and you know, they're shining them, and to me it just like makes me really happy to see, but it shows like the dynamics between societies and the environment, because this is something, you know, that was farmed, and then there was a hurricane in the 60s that completely wiped out agriculture for the most part on the island, and things have kind of never been the same since. But it does have this huge cultural significance. And you know, this isn't something that's important. They're shining the beans, so it's not something that's essential to their production. It's just something that, you know, makes them more valuable, but it's not essential. It's just like something that people like to do, and it has this like strong historical significance. So that was it. Thank you all for listening. A little bit over the place, but I just thought I'd share a little bit about what I do.